The overall goal of this procedure is to generate highly pure sinus nodal cells. This is accomplished by first inducing cell differentiation by modifying the medium and making hanging drops that result in the formation of embryonic bodies of a defined size. Next, the embryonic bodies are seated on a gelatin-coated dish so that they form a cell layer. Then the antibiotic selection is initiated and only cardiomyocytes survive. Finally, the cell layer is dissociated to obtain fast-beating single sinus nodal cells or small sinus nodal bodies. Ultimately, live cell imaging is used to show high beating frequencies of sinus nodal cells.
The main advantage of this method is that you can produce highly pure sinus nodal cells, for example, for drug testing.
The method can help us answer key questions in sinus node disease because the cells will give us insights into the underlying mechanisms. The technology will be presented in the lab by Jung, our PhD student, who developed the method.
Beginning with an ES cell line and pluripotent stem cells co-cultivated with irradiated feeder cells, as outlined in the text protocol, 2 days before the differentiation procedure, remove the PSCs from the feeder cells by first aspirating the medium. Use 10 milliliters of PBS to wash the cells, then add 7 milliliters of collagenase 4 solution and incubate the cells for 10 minutes at 37 degrees Celsius. Next, place a sterile 40 micrometer filter in a 50 milliliter tube. Then carefully